I don't know about all of you, but I'm so ridiculously jazzed right now because this is our very first fully vodcasted lecture, and also because it is Friday. And if you're watching this from the regularly scheduled lecture, it is Friday, October the 24th, and you can get stoked because that means that next week in lab, you will be getting your unknown bacteria. So if you haven't had a chance yet, you're going to want to make sure that you are fully ready to profile unknowns, that you are fully aware of what the characteristics of certain criminal bacteria are so that you can do a good job of identifying the culprits that you might have. Now, not in all cases are we looking for criminal bacteria. Sometimes we're looking for really rad, awesomely helpful bacteria. Maybe we're trying to identify certain soil microbes that do their jobs in perhaps bioremediation or what have you. So there are many different ways and reasons that you might want to profile unknown bacteria. And so this is something that we need to be ready to go to do next week in lab. So if you have a chance this weekend, spend a little time dorking out with those biochemical tests. Remember that online there is a source available that is a summary of biochemical tests. It's a photographic resource. It's under additional information on the lab webpage. So find that, check out the pictures, dork out with the pictures. It's not too nerdy to add them to your cell phone as a cell phone background. It's a good thing to do to get ready to go, to know that you will be able to identify unknowns. Now, if you haven't already, this is the last day to turn in your skill check in lab. So make sure that if you haven't given that to your TA, that you do before the end of today. So make sure that it is turned in. It is due today, Friday the 24th. I would like to enter midterm scores very soon for all of you. And so please make sure that your grades on YO courses look like they're up to date and that they represent your effort at this point. Remember that midterm grades come out soon. They're just whether you're passing D or F, and so you want to make sure that's representative of what your current work is. I am most likely, even as we speak, still working my way through the hypothesis revisions and honestly enjoying them, but I have not quite completed them, and when I do, I'll make sure that you get feedback on those. Homework 6 is due on Tuesday, and get jazzed because we have started a new fevered discussion, and I hope that it will take off and get running hot as soon as possible. That being said, we are going to hop right into gene expression. We're going to pick up where we left off last time. So recall that in replication, we were looking at the riveting process whereby DNA polymerase was very, very rapidly and accurately synthesizing DNA corresponding to the template strand. And we were seeing that bidirectional and semi-conservative process leading to the duplication of, for example, the chromosome of a bacterial cell. We looked at E. coli. Today we're going to turn our attention and look at the process within, again, situating ourselves largely within prokaryotes, but looking at the process whereby the DNA is converted into RNA code. So this process is termed transcription. So our goal then is to really zoom in and highlight all of the features and special differences about transcription, what it does. I want to take a minute to make sure that everybody understands that upon transcription, there are 3 possible products that are made. mRNA, tRNA, and rRNA. Now, tRNA and rRNA do their job in the cell as RNAs. That is, they do not go on to be further translated into a protein product. But what's cool about them is that they both do their job helping mRNA to get converted into protein products. So tRNAs and rRNAs are the workers of translation, and they do their job as RNAs. So let's go ahead and talk about the process by which these RNAs are synthesized. And the first thing that we have to do is a lot of lingo. There is a lot of vocabulary that surrounds the process of transcription. So let's situate ourselves first looking at a gene. That is the region of DNA that is going to get converted into product. whatever that product may be. And we always orient ourselves in the same way when looking at a gene, that is just through tradition. We look at it with the 5' to 3' strand being the top strand, and by convention this strand is called the coding strand or the plus strand, and it's always that 5' to 3' orientation. That is, if you were to look at the end of the DNA, you would see the 5' phosphate on this side on the left and the 3' hydroxyl on the right. Of course, things are a little bit more complicated when you're looking at a plasmid because it is completely circular. So we'll talk about how, in fact, we can orient ourselves on plasmids too. So notice then that the opposite strand, of course, will be anti-parallel to the coding strand, and it is the 3' to 5' strand. And as you might guess, instead of being called the plus strand, it is called the minus strand. So the top strand, the plus strand, the coding strand, the 5' to 3' strand, all of these are terms for that top strand, whereas the bottom gets some of the opposite nomenclature. It is the 3' to 5' strand, or the negative strand, so it's the anti-parallel strand to that top strand. So template is also the term given to this bottom strand, and that's because it is this bottom strand that serves as the code to make the RNA. That is, it is the template for the synthesis of the RNA. I'm going to show you that in just a moment, but first I want to make mention that the gene by definition is always termed the +1 nucleotide. That is, the start site for the gene is always given that +1 term, and then the ending site is the termination sequence. So we'll be seeing that as standard nomenclature no matter what gene you're looking at. So the gene always begins at what we designate the +1. So then it means that the nucleotide to the left of that +1 nucleotide is the -1. There is no 0 by convention. And then to the right of that nucleotide is the +2. So we can talk about all of the nucleotides in a gene. We could talk about the 684th nucleotide based upon the +1 being the start site. So that's a really important set of terms that goes with this. So as I said, the template strand is given its name because it is what serves as the guide to make the RNA. And I think that this is going to be something I'm going to say this like a bazillion times because I want to make sure everybody is really understanding it. But I'm going to actually draw it, and I'm going to do a lot of drawing today because I think it'll help you to visualize all that is going on with transcription. So I'm going to make a little drawing down here showing the same thing that we have up here, so that I can give you a better feel for what happens as this DNA is converted into RNA. And I promise we're going to do this about 10 more times at least before it's all over, because I have a feeling it's the beginning of the hour right now. Everybody's still asleep. I'm going to wake you up a little bit, and then by the end of the hour, hopefully all of this will start to make sense. So we've got our coding strand on the top, the red one there. We've got our template strand on the bottom, the blue one there. And what is going to happen is the enzyme that reads the bottom strand is going to make the RNA, and that enzyme is very fittingly called RNA polymerase. Remember how DNA polymerase made new DNA? Well, RNA polymerase makes new RNA. So we're going to see RNA polymerase come on in, and it's actually going to bind at this region that I've shown in blue over here. So we can kind of make ourselves a little blue box, knowing that the enzyme that reads the template strand is going to bind at that blue box. So we'll make this enzyme black. And when this enzyme actually binds to the DNA, it actually serves to melt a stretch of that DNA. So I'm going to try to show you that a part of the DNA, that the hydrogen bonds between the strands are broken apart, and we're going to see it melt. So there's going to be this little melted region, this little bubble. And that actually, I know it doesn't sound like a technical term, but that actually is totes a technical term. So we're going to see this bubble called transcription bubble. And the RNA polymerase did it on its own. So remember how we talked about DNA polymerase requiring DNA helicase to unzip genes? Well, RNA polymerase can unzip its own genes, right? It can do it itself. And it's going to head on down and read the DNA. So as this RNA polymerase enzyme reads the DNA, it's going to read the template strand. So I'm going to do a quick time lapse of this, showing that after a certain amount of time, and I promise we're going to do this again, you're going to be totes bored with me by the end of the hour, especially because I keep using the word totes. Credit to my skiers. They get me in that habit. I once read an article, some sort of blog post that said people over 30 shouldn't use abbreves. And so I decided then I must not be over 30. Okay, so what we see here is that after time lapse has occurred, now this RNA polymerase enzyme is further down reading the gene, and it's reading this blue strand. So as it reads, it's pairing RNA, and I'm just going to make the RNA the same color as the coding strand because the RNA is pairing transiently. It's hydrogen bonding right here temporarily and transiently with the coding strand. So what this means is that the RNA that's getting made by the RNA polymerase enzyme looks exactly like the coding strand, the top strand, only it's RNA, not DNA, right? So all of you will tell me, based on your awesome performance on the first exam, that means that U's replace T's everywhere there was a T in the coding strand. They become U's in the RNA. And of course, in all cases, it's a ribonucleotide instead of a deoxyribonucleotide. So we recognize that that would be different in the composition. But otherwise, this looks like, and in fact, this is, let me do this in red, this is the 5' end of the RNA. So very much like the DNA, it looks almost exactly like that coding strand because it paired with the template strand, right? Everywhere there was a G, it paired with a C. Everywhere there was a T, it paired with an A. Everywhere there was an A, it paired with a, almost tricked you, a U, right? And that gave rise to the RNA. So let's make sure that, again, in case this still doesn't make sense, I promise you 10 more times I'm going to cover it because it's probably like one of the most important things that we cover in microbiology. So the RNA has the same sequence as the coding strand of DNA, except for that U's replace T's. So what that means is that if you're ever given the sequence of the coding strand of DNA, it's very simple to write the RNA sequence. You just write U's everywhere that you see a T. Awesome. Let's continue on with our vocabulary. So notice that blue box is where I said that the enzyme RNA polymerase binds and begins the process of transcription. It's not actually a big blue box, and it's a sequence of nucleotides called a promoter. This promoter, I was thinking about it, and I've got skiing on the brain, of course, and I was thinking this is a lot like the start line of a race. The promoter is where everything assembles, and say you're a skier getting ready to start your race, you're going to need a lot of things to help you with that race. That is, you're going to have your poles and your skis and your race suit, and the same thing is true of RNA polymerase. RNA polymerase is going to need to be assembled right there. It needs to have special factors that allow it to recognize the promoter, and it's got some things called transcription factors that associate with it. So it's like everything comes together right there at the promoter or the start line for the process of transcription. So an initiation site at which the transcription complex assembles. And what's interesting is that in bacteria, there may be a single promoter that controls many, many genes. So we may see more than one gene controlled by a single promoter. And I'm actually going to make a drawing of this. Like today is totally drawing day. And we're going to see in some bacteria, some archaea, we might see a situation where we don't just have one gene, but instead we have many genes that are all controlled by the same start line. And this is actually going to matter. So if this is our promoter, I'm going to put P for promoter. We might have, maybe 5 genes that are all controlled, and I'm just going to number them 1, 2, 3, 4, 5. Wow, this is a lot of genes, all controlled by that same promoter. So what that means is that when RNA polymerase binds at that promoter and it begins reading the genes and it reads the template strand, well, then that means that it's going to make an RNA that has the information for all 5 genes on it. So let's say time has passed here, and now we get the synthesis of an RNA, and that RNA, of course, looks like the coding strand, only U's replace T's, so we're going to get the RNA, but on that RNA, we're going to have information for gene 1, information for gene 2, and I'm getting a little ahead of myself, so information for gene 1, information for gene 2, information for gene 3, 4, and 5, all on the same RNA. That RNA, of course, looking like the coding strand. So this is cool because what this means is what is made is what's called a many polycistronic mRNA. So when we see more than one gene under control of the same promoter, we actually call it, so this is called an operon. We call it an operon. And the mRNA that's made is called polycistronic. The information for many genes is kept on that RNA. So anytime you see something that looks like this, where there's more than one gene controlled by the same promoter, you can quickly say to yourself, "Ba-bum, that's an operon," right? So we recognize that as being lingo. Now, if we see an mRNA that has the information for multiple genes, we can quickly say, that's a polycistronic mRNA. Now, in eukaryotes, generally every single gene has its own promoter. So generally every gene has its own promoter. mRNAs carry the information for only one gene, and as you guessed it, they are not polycistronic, they are monocistronic. Once again, a little more boring than our bacteria and archaea. So as we mentioned, the promoter is not literally a blue box, and in fact it is a unique sequence of nucleotides, and I want to investigate now at this point what that unique sequence is. So hopefully everybody's got this drawing down. If not, pause me and get your drawing done and get your thoughts collected. And I want to go ahead and talk about what these promoter sequences are. So every promoter is defined by a consensus sequence. That is, if we take all kinds of different promoters and we compare them, we can find out what nucleotide happens in each position the most frequently. And that allows us to identify what we would call an ideal consensus sequence. That ideal consensus sequence includes 2 areas. There's a region 35 nucleotides to the left of the start site. That is, if you remember, if you start, the +1 is the start site, -1, and then you count all the way to -35, you're going to hit the first consensus sequence. So of course that's in this region that we're showing as the blue box. That first -35 sequence is very surprisingly called the -35 sequence, and it is always ideally TT GACA. TT GACA is the ideal consensus sequence. That doesn't mean that promoters always have exactly TT GACA. It just means that they're the strongest, or they're the most frequently transcribed when they do have that ideal sequence of TT GACA. But of course the -10 region also contributes to this. So a little closer to the start site is the -10, or the Tata box. The Tata box is by far the most famous of promoters because not only do bacteria and archaea have a -10 Tata box, but we also see a Tata box in eukaryotes. It's in a different place, but it still is a Tata consensus sequence. So if a bacterium has a TT GACA Tata at sequence that is very, very similar to this sequence, then we say that it is a strong promoter and it's transcribed really, really frequently. That is, it closely resembles this ideal and it makes it very frequent to be transcribed. If it's actually really a poor match, like maybe it's TACACA, then it's much weaker. That is, there are less of these nucleotides that match the ideal sequence, and we would say that the promoter is weak and it's transcribed infrequently. Now, the question does present itself as to whether it's always good to have a strong promoter. A strong promoter means frequent transcription, but not all genes, it's not beneficial for all genes to be transcribed super frequently, right? Because some gene products aren't needed in high concentrations. So one way to regulate the expression of genes is to change what that consensus sequence is, and then cells actually use that to their advantage. It allows them to determine the level to which those gene products are expressed, right? Maybe sometimes it's beneficial to have only a trickle of expression, whereas sometimes it's, for certain gene products you want a downpour, and some of those would be gene products that are associated in day-to-day housekeeping functions. Those are going to have very strong promoters, whereas those that are needed less frequently are going to have very weak promoters. So it really does depend on what the gene product does within the cell. Let's practice and see if we can pick out the promoter that is the strongest. That is to say, the one that has the sequence that is most similar to ideal. So starting with the top one there, let's see if we can highlight the differences. So GACA, there's the first difference. That would be a GACA. Okay, so there's only one difference there. TATA, two differences there. Okay, so oh boy, two differences already, GACA. TATAA, wow, that one's a weak promoter, right? Gaga, okay, there's only one difference. Oh, there we go. There's our strongest promoter. There are only two differences right there from ideal. So Gaga, that is our strongest of the three promoters. Since we're so on fire right now, and I don't know about you guys, but I'm totes feeling jazzed. So let's go ahead and go on and talk about another example where we can apply this same concept. Not only do we get to apply the concept here, but we also get to review. So you get to flex your mind on the whole replication process that we already covered. So all of the following accurately describe either prokaryotic replication or transcription except. So notice that we're looking for the false statement. During replication, Okazaki fragments are formed during the synthesis of the lagging strand, but not during the synthesis of the leading strand. Absolutely, right? That's the whole Okazaki fragment thing. That's what that's all about. It's only done when their lagging strand is synthesized discontinuously. The next deoxynucleoside triphosphate that DNA polymerase would add to the currently synthesizing strand would be DCTP. Okay, well, remember DNA polymerase is wicked fast, wicked accurate, but it's very needy, right? It needs a lot of things, and one of the things that it needs is an open 3' hydroxyl. So look at here, we have one open 3' hydroxyl here, and one here, okay? But the other thing that DNA polymerase needs is a template, right? It needs building blocks and a template. So if we assume building blocks are present, only in one case do we see actually a template. Notice that right here, if cytosine tried to pair with something, there isn't a template to be had here, right? There isn't another strand to guide what would be added at this 3' hydroxyl. How would the polymerase enzyme know what would go next? Because there isn't any template here. However, if we look at this 3' hydroxyl, notice that guanine loves to pair with cytosine. So it would pair with a C right there, and the next nucleotide that would be added right here would be DCTP. So let's see how that sounds. Yes, good. Okay, bing. That one is checked off the list. True, because we're looking for the false statement, right? So true, true. And then down here, a promoter with a -35 sequence of TGCAGA. Oh boy, okay. TTGAGA. Oh man, that's not very strong, is it? And a -10 of TATAT, there's our differences, is stronger than a promoter with -35 of TTGA. Oh, one difference. And oh no, that is a false statement. because clearly the first promoter is weaker than the second promoter. So we've got our answer, but let's see why D is true. The coding strand of DNA has the same sequence as the RNA transcript, only U's replace T's. Bingo! The concept I said I would cover at least 10 times today. So right there we've hit time number 2, and I promise you at least 8 more times.